Hello, Professor. Today I'll be talking about acetylcholine, the signaling between ganglionic neurons and the receptors on the ganglionic neurons. So first, just to tell you what ganglia is, it's a group of cell bodies located within the peripheral nervous system. A ganglionic neuron or a preganglionic neuron is just the nucleus from which the axon leads to the ganglia. At the terminal of the preganglionic neuron, there are vesicles rich in acetylcholine. These terminals also contain voltage-gated calcium channels. When calcium rushes into the terminal, it stimulates the migration of synaptic vesicles to the membrane, triggering exostosis of acetylcholine. And the acetylcholine will go to the post-ganglionic neuron receptors. And the receptors on this that I'll be talking about in this video are the first one I'll be talking about is nicotinic receptors. There's two types of nicotinic receptors, and then nicotinic receptors are present on neurons and found at the autonomic ganglia. And then nicotinic receptors are present on skeletal muscle at the neuromuscular junction. Nicotinic receptors are ligand-gated ion channels, and when the acetylcholine binds to the nicotinic receptor on the postsynaptic membrane, the channel opens, allowing sodium ions to enter the cell. This depolarization leads to the opening of voltage-gated sodium channels, which generates an action potential. At the neuromuscular junction, the NM nicotinic receptors, these contain two acetylcholine binding sites. When acetylcholine binds, sodium will enter the cell and produce a motor end plate potential. If threshold is reached, then voltage-gated sodium channels open and an action potential is produced within the muscle cell. So now I'll move on to the other receptor, which is muscarinic receptors. Muscarinic receptors respond to acetylcholine and muscarine. These receptors are known as G protein coupled receptors. And there are 5 different types: M1, which stimulates, M2, which inhibits, M3, which stimulates, M4, which inhibits, M5, which stimulates. Positive muscarinic receptors, which are the ones that stimulate, operate through the GQ protein pathway. When acetylcholine binds the receptor will activate a G protein that exchanges a GDP for a GTP. This activates phospholase C, which is an effector enzyme, which breaks down PIP2 into signaling molecules. One of these molecules stimulates the release of calcium from the endoplasmic reticulum into the cytoplasm. Calcium then activates enzymes that work with the protein kinase C to phosphorylate specific proteins that the cell may need. Negative muscarinic receptors, which are the ones that inhibit the function through the G inhibitory pathway. When acetylcholine binds, the G inhibitory protein becomes activated and splits into subunits. The beta and gamma subunit binds to potassium channels, that are leaving, which produces a hyperpolarizing effect. The alpha inhibitory subunit binds to adenylate cyclase, which is another effector enzyme, and this prevents the conversion of ATP into cyclic AMP. And finally, we're talking about the location of the receptors. M4 and M5 receptors are only found in the central nervous system. They're not as common as M1, M2, and 3. M1 receptors are commonly found in the central nervous system and in parietal cells within the stomach, where they trigger the release of hydrochloric acid and some pepsinogen. M2 receptors are found in the heart on the SA node and the AV node. They are also present on the presynaptic membrane, where they inhibit the release of acetylcholine. M3 receptors are found on exocrine glands and on smooth muscle. They're also found in the lungs, which creating bronchoconstriction. They're found in the gastrointestinal tract. They're responsible for bladder constrictions, defecation, and the near vision and narrowing of the pupil effects in the eye. That is all I have for this video. Thank you for watching.